From this article, I think the biggest thing was cytochrome P450s, or also known as CYP enzymes, are probably one of the top causes of drug interactions. And it can get quite complicated. Drugs can be CYP substrates, they could be inhibitors or inducers, or actually a combination of both. And that can significantly impact the way drugs work in our patients. And so knowing not just the type of CYP enzymes, but the complexity of the isoforms in humans versus veterinary species. Like in humans, like CYP3A4 is common, but the equivalent in dogs is CYP3A12 or CYP3A131 in cats, and how those drugs can be very different when you're going through those CYP enzymes. So veterinary specific studies can play a huge role in helping us understand and navigate all these different types of interactions to provide the optimal drug therapy for our patients and minimizing adverse effects with these combination of drugs that go through the same CYP pathway.
I think in addition to minimizing the adverse effects, another thing that I found really practical was sometimes we actually use these interactions to our advantage. And I know certainly I've done that multiple times in my career, pairing like fluconazole with cyclosporin, and that way I can reduce the costs for the owners and the dose and maybe adverse effects of one of the medications. I thought another really cool example was pairing fluconazole with oral methadone, so that we can actually prolong methadone's analgesic effect. And so it's nice to remember that we want to avoid problems, but we can also make smarter therapeutic choices using these interactions.